Okay, just before we call Dr. Pyke to come speak, I want to invite everybody in the room just to take a quick second, maybe stand up, stretch your legs if you'd like, because it says physical activity people. You're going to feel better, and Dr. Pyke will be able to explain to you why getting the blood flowing is making everything
true exercise physiology.
So, since you've already started with the standing ovation, I will introduce Dr. Kyra Pyke. It really is my honor to introduce Kyra. Kyra's come to us from Queen's University, where she's an associate professor in the School of Kinesiology and Health Studies. She did her undergraduate degree at York University in kinesiology, her graduate training in exercise physiology at Queen's. She's a recipient of an early research award from the Ontario Ministry of Research and Innovation and the SGPS John G. Freeman Faculty Excellence Award. This is all just off the top of my head. Dr. Pyke's research investigates three interacting areas of human cardiovascular research. The first being endothelial function, the second being autonomic function, and the third being physiological effects of psychosocial stress. Her laboratory focuses on improving our understanding of how the endothelium responds to changes in blood flow, advancing endothelial function assessment in humans, and understanding how endothelial function and arterial stiffness change with exercise, psychosocial stress, and disease. On a personal note, Dr. Pyke's lab has certainly supplied me with some great trainees, including a current postdoc, and we've had many trainees who have gone on to do bigger and better things in Kyra's lab as well. So, Kyra, I'd like to present you with this award as a distinguished speaker, and thank you again for coming to us. Dr. Pyke's talk today will be titled Conduit Artery Endothelial Function. Are two tests better than one?
Okay, hello everyone. So first I'd just like to say what an honor it is to have been invited to come and speak today. I'll thank Dr. Tammy Martino and everyone who's been organizing this. Has been wonderful. Josh, who's been my tour guide and kept me safe around campus all day. All the people who I visited with, it's been really great. And so to begin here, I'll just give you a little outline of where I'm going to go. So I'm going to start by just reviewing some important functions of the endothelium, and then I'll talk about how conduit artery endothelial function is very typically assessed in humans. So there's sort of one index of endothelial function that gets the most attention. And then for the rest of my talk, I'll focus on some recent findings telling us about the utility of maybe using an additional index of endothelial function to tell us about how the conduit artery endothelial function works. So to begin with, just a little schematic of a blood vessel, and the endothelial cells are right here lining the lumen of the vessel, so they're in direct contact with the blood. And the endothelial cells have vasoactive and vasoprotective properties. And so by vasoactive, I mean that they help us to control vascular tone. And by vasoprotective, I mean that they exert a whole array of functions that can help to inhibit the development of atherosclerosis. And one important molecule there is nitric oxide, which can be produced inside the endothelial cells by endothelial nitric oxide synthase. And a lot of the vasoprotective function of the endothelium is attributed to what nitric oxide can do. And indeed, nitric oxide is a vasodilator, and so the vasoactive and the vasoprotective functions of the endothelium are linked. So why do we assess endothelial function? So as the story goes here, if we have endothelial dysfunction, that will result in an attenuation of vasoprotection, and so we might have increased vulnerability to atherosclerosis development. So if we're able to measure endothelial function early on, we may be able to predict the risk of development of cardiovascular disease. In addition, because endothelial function is involved in controlling vascular tone, if we have changes in endothelial function, that could tell us about problems in the regulation of vascular tone that could lead to things like difficulties with perfusion or regulation of blood pressure. So knowing about endothelial function can give us some mechanisms of what might be going on with changes in the control of vascular tone. So how then do we assess endothelial function in humans if I've convinced you that this is something that we want to do? There are a whole number of different indices of endothelial function, and I've divided them into two sort of broad categories here. We can assess factors that are circulating in the blood, or we can assess endothelial dependent dilation. and there are a couple of ways that that can be done. And so if we really think about endothelial function or functions comprised by this whole pie, each one of these indices probably only captures sort of part of endothelial function, okay? And what I'm going to be talking about today specifically are endothelial responses to blood flow associated shear stress, and even more specifically, I'm going to be talking about the conduit arteries. So to do so, I've got a little model here. This would be a little bit of lumen of a vessel with, this is where our blood is going to flow. We then have an endothelial cell and then an adjacent smooth muscle cell. So when we have an increase in blood flow through our arteries, that results in an increase in shear stress as well, that sort of mechanical frictional force against the endothelial cell. Now, you may hear me say shear rate as well, and that's simply an estimate of shear stress without accounting for blood viscosity. So I may use those two terms interchangeably. So when we have an increase in blood flow and shear stress that is sensed by mechanosensitive structures on the endothelium, and then that results in signaling that ends in the production of a number of different vasodilators by the endothelial cell, and one of them, nitric oxide, we've already talked about. These can then act on the vascular smooth muscle, cause it to relax, and then that's our vasodilation. And we can measure these changes in arterial diameter as the index of endothelial function. So larger change in diameter is indicative of better endothelial function. And as a whole, this is called flow-mediated dilation because increases in blood flow result in increases in shear stress. And you'll hear me shorten this very frequently to FMD, flow-mediated dilation. So how then do we assess FMD in humans? Basically, we need to increase shear stress to create the stimulus and then measure this change in diameter. And this can be done in the coronary arteries, which is of interest because if we have endothelial dysfunction, and that does increase risk of atherosclerosis. Atherosclerosis, of course, in our coronary arteries contributes to cardiovascular events like myocardial infarction. But assessing endothelial function or flow-mediated dilation in the coronary arteries is an invasive endeavor that requires catheterization, infusion of drugs to create the shear stress stimulus, and dyes to enable imaging of the arteries. And we often want to assess endothelial function in people who are currently healthy because we want to know about the dysfunction before the disease becomes clinically manifest, and because we just want to know about mechanisms of vasodilation. And doing these very invasive procedures isn't always appropriate in people who are currently disease-free. So how do we deal with that? We can assess endothelial function and flow-mediated dilation specifically in peripheral arteries, and that can be done non-invasively. So I'm showing the most typical setup here. If you forgive my little schematic, this is our brachial artery, not just a red rectangle. And this is an ultrasound probe that is sitting over the brachial artery, and we can get an image with ultrasound of that artery, and then we can also, through Doppler, get the blood flow velocity signal, and from that we can calculate the shear stress. And so we need to create a shear stress stimulus if we're going to measure FMD, and to do so we need to reduce the downstream resistance, so the resistance in these vessels downstream from the brachial artery. And by far, the most common method that's employed in humans to do this is to create a reactive hyperemia. So an occlusion cuff is put around the forearm at a suprasystolic pressure, inflated for 5 minutes. This results in a downstream dilation, so a reduction in resistance. Then the cuff is released, and we have a reactive hyperemia. So blood flow and shear stress increase through the brachial artery. And I'll just show you a little schematic there. This would be our shear stress stimulus upon cuff release, big increase, and then it quite quickly subsides. So this shear stress stimulus might only last 30 to 60 seconds. So it's quite transient. This shear stress is going to stimulate flow-mediated dilation in the brachial artery, which again we can also measure with ultrasound, and it's this peak percent change or just peak change in diameter that's used as our index of endothelial function. And because I'm going to talk about another FMD index, I will refer to this technique, which is again really the standard dominant technique, as reactive hyperemia FMD or RHFMD. And I'll even use this little cuff symbol when I'm referring to RHFMD so that you can distinguish. Okay. So RHFMD, there is a relationship between the FMD that happens in the brachial artery and then FMD measured invasively in the coronary arteries. And so that's shown in these coronary artery disease patients. And so that means that brachial artery FMD is often used to sort of reflect what's going on in the maybe more clinically relevant arteries in the heart. In addition, reactive hyperemia, FMD here, can predict cardiovascular events in people with CAD and also people who are currently healthy. So all this sounds great. Is RHFMD all we need to characterize conduit artery endothelial function in humans? So maybe yes, right? So it can be performed non-invasively, it reflects coronary artery endothelial function, which is clinically relevant, and it's an independent predictor of cardiovascular risk. But my presentation's not over, so maybe no. And I'll go through 3 points here to suggest that maybe looking at another index may also be helpful. So the first point that I will mention is that the reactive hyperemia stimulus is uncontrolled. So what do I mean by that? Here's my little schematic of the reactive hyperemia stimulus again. So this would be cuff release over time. We have shear stress on the y-axis. And the stimulus is really quite variable. So when you release the cuff, you just get what you get. And some people will have a much larger stimulus than others. And even within a person, if you do repeat trials, you'll see a difference in the magnitude of the stimulus. There is a stimulus-response relationship. So if this is a range of shear stress stimulus magnitudes here on the x-axis, and this is just expressed as the area under the curve of this whole shape here, as the shear stress stimulus increases, we'll see a larger RHFMD response. So if we just imagine for a moment now that the people who had the small stimulus are in one group, and the people who had the big stimulus are in another group. And so if we were to just plot those means of RHFMD, we might get something that looks like this. So the group that experienced the small stimulus had a small RHFMD, and the group that got the big stimulus had a big RHFMD. Now we want to be able to conclude that a small RHFMD is indicative of endothelial dysfunction. That's what this index is for. However, we can see because of this dose response or stimulus response relationship, the picture is not so clear because we don't know whether these people in group A do indeed have endothelial dysfunction or if they just experienced a small shear stress stimulus. And that can be quite difficult to tease out. And this is a problem that still sort of plagues the use of RHFMD, even though it is very, very frequently used. So this issue of stimulus control does not plague animal studies that look at FMD. So there are long-standing sort of preparations that afford excellent control of the stimulus. So this is just one example. We've got shear stress here, which is a sustained and controlled shear stress. And then we have a flow-mediated dilation response. And this kind of data just really wasn't available in humans when I started to become interested in looking at endothelial function. And that was actually sort of one of my earliest motivators, was wanting to be able to examine endothelial function in humans using the same level of shear stress stimulus control that's been available in animal models. And there are a number of ways to do that, although surprisingly very rarely adopted. There's only sort of my group and a couple of others that use sustained increases in shear stress to look at FMD in humans with any sort of degree of regularity. You can create a sustained increase in shear stress in a number of different ways. You can heat the arm, you can infuse vasodilator drugs. I'm going to talk about the model that we've really settled on and used quite a bit in my lab, which is using exercise to create the shear stress stimulus. So in our model, we use isometric hand grip exercise. This is a little picture of a dynamometer. And with isometric muscle contraction, we're going to dilate the muscles in the forearm, and that's going to increase blood flow and shear stress through the brachial artery. And the increase in shear stress will simply vary with the exercise intensity. So bigger increases in shear stress with greater exercise intensity. And so when I talk about this FMD, so FMD that's stimulated by these sustained increases in shear stress, I'm going to call that sustained stimulus FMD, or SSFMD, which is a term that we sort of coined in a recent review article. So what kind of control and what kind of shear stress stimulus profiles can we create using exercise? So this is data from Dr. King, who is back here in the audience. He is now a postdoctoral fellow, but this was way back when he was doing his MSc with me at Queen's. And so what we looked at was, we looked at 3 different conditions where we were comparing different exercise duty cycles. So simply different ratios of duration of contraction to relaxation. And what I want to point out is that we were able to really achieve the same shear stress stimulus in all 3 of those conditions. And then beyond that, between subjects, we were able to achieve the same shear stress stimulus. So you can see these really narrow error bars. So in all of our participants, we were able to get really a uniform shear stress stimulus magnitude. And that's because we're able to target a shear stress stimulus using exercise. So how do we do that? Once we select our shear rate target, in this case, we chose 75. We combine that with knowledge of, in this case, the brachial artery diameter. And then we simply rearrange the shear rate equation to calculate a required velocity to hit our shear rate target. And so in this case, that required velocity would be 26 centimeters per second. And our system allows us to see on the fly, when someone is doing this isometric exercise, what their blood flow velocity is measured with ultrasound. And so if we're not hitting our required velocity, we simply have them contract a little bit harder. If their velocity goes too high, we just have them contract at a lower exercise intensity. So we're really able to maintain this target shear rate. And that can be used not only to create these sort of step increases in shear rate, but you can create incremental increases in shear rate as well. This is work by one of the few other groups that uses this model out of Utah. And so one question that we had and that reviewers had when we started using exercise to increase shear stress to examine FMD was whether, when we were creating these increases in shear stress with exercise, it was really all shear stress mediated dilation that we were observing in the conduit artery. Because of course, when we do exercise, we increase, oh, that's bad. There we go. We increase shear stress, but we also have metabolic dilation happening in the microvasculature of the forearm. And some of that vasodilation, there are a number of mechanisms by which that dilation can conduct cell to cell. And so the question was, is some of the dilation that we're measuring in the brachial artery actually conducted vasodilation and not only an FMD or shear stress mediated mechanism? And so to figure out if that was so, we had participants perform hand grip exercise at the same intensity that they did over here that elicited this magnitude of dilation in the brachial artery. So we had our metabolic stimulus for conducted vasodilation, but then we applied arterial compression. And this can actually be done just with finger pressure on the brachial pulse at the antecubital fossa. You can really control the shear stress stimulus in the brachial artery. And so with this arterial compression, we were able to do the exercise and have the metabolic stimulus, but eliminate the increase in shear stress. So there's now no stimulus for FMD in the brachial artery, and we saw that that also eliminated the brachial artery dilation. So there was no dilation, in contrast to the situation here where there was a pretty big dilation when shear stress increased. So to put that together then, conducted vasodilation doesn't appear to reach the level of the conduit artery in this hand grip exercise model at least. And we know that because when we prevented an increase in conduit artery shear stress, we also prevented the increase in conduit artery dilation. And so that then allowed us to sort of move forward, knowing that when we were using exercise to create our shear stress stimulus, we were indeed measuring FMD in our conduit artery. And so we might ask then, what mechanisms are responsible for SSFMD? So with RHFMD, there's been quite a bit of work interrogating the nitric oxide dependence of RHFMD, because again, nitric oxide is a vasoprotective molecule. So once again, this group in Utah, Ray and colleagues, investigated the nitric oxide dependence of SSFMD stimulated by hand grip exercise. So they used an incremental exercise model with saline infusion or infusion of the nitric oxide synthase inhibitor LNMMA. And they found that like RHFMD, SSFMD was significantly nitric oxide dependent. So both indices of FMD seem to have a significant nitric oxide mediated component. And so what can we do with SSFMD, right? We wanted to be able to create a model so that we could control the shear stress stimulus to do different sorts of investigations that we can't do when just using an uncontrolled transient stimulus with RHFMD. And so we can do lots of things, but I'll just show you one example here. In this study, we were interested in looking at the impact of the rate of increase of the shear stress stimulus on the dilation response. So we created a step increase in shear stress in the brachial artery, and then a ramp, a 5-minute ramp increase in shear stress in the brachial artery. And then when we look at the dilation response, we saw that once we reached the same steady state shear rate, which we're doing by about minute 6 or 7 here, we saw the same magnitude of dilation in both conditions, which tells us that it's really the magnitude and the duration of the increase in shear stress, which is determining the SSFMD magnitude, and not the initial rate of onset of that shear stress stimulus. And we were motivated to investigate this because this type of manipulation has been available in animal models for quite some time. So this is data from 2000, and they performed the same manipulation in rat arterioles. So we've got a step increase in shear and then a ramp increase in shear. And we can see that the dilation pattern is really quite different in these animal arterioles compared to what we saw in humans. Okay, so is RHFMD all we need? Maybe not, because that RH stimulus is uncontrolled, and SSFMD models can provide us with stimulus control that allows us to sort of ask and investigate new questions about endothelial function in human conduit arteries. Another reason that we may have interest in investigating SSFMD is because it's a physiologically relevant stimulus. And so by that I mean it happens in our daily lives. So SSFMD happens. When we are hot, we have sustained dilation of the microvasculature, so sustained increases in shear stress in our conduit arteries, so too with exercise. So if we want to understand how our conduit arteries are sort of functioning in normal daily activities, it's important that we investigate SSFMD. And then finally, endothelial transduction of transient versus sustained increases in shear stress appears to be at least somewhat distinct, suggesting that these two different shear stress stimulus profiles, when we measure the FMD, it's telling us about somewhat different aspects of endothelial function. So I'll just show one bit of cell data. So this is the shear stress magnitude put onto cells that are plated, and those cells were exposed, and these are human umbilical vein endothelial cells. They were exposed to an impulse or transient shear stress stimulus and then a sustained shear stress stimulus. And it was found that the endothelial cell NO production in response to that transient shear stress stimulus was dependent on G protein signaling, whereas the nitric oxide production by the cells stimulated by the sustained increase in shear stress was independent of G protein stimulation, or signaling, sorry. And further data has illustrated that transient increases in shear stress stimulate calcium-dependent nitric oxide production, while sustained increases in shear stress stimulate nitric oxide production by endothelial cells that's independent of calcium signaling. So taken together then, these signaling transduction pathways that happen when shear stress increases seem to be at least somewhat distinct, depending on whether the endothelial cell has experienced a transient increase in shear stress or a sustained increase in shear stress. And so that would suggest, again, that if there are these distinct shear stress transduction mechanisms, that then RHFMD, which is exposure to a transient increase in shear stress, and SSFMD, which is exposure to a sustained increase in shear stress, might interrogate different aspects of endothelial function. And so the way that we've looked at that is by comparing RH and SSFMD in response to, or in people with different chronic conditions and in response to acute interventions. So I'll just show you a little bit of that data. So this was us looking at young, healthy males who were smokers. They had a short history of smoking, or controls who were non-smokers. And we did our RHFMD test and a hand grip exercise SSFMD test in both groups. And we can see that the stimulus magnitude was the same. So we were able to, the groups were not different in terms of the shear stress stimulus. What about the FMD response? So RHFMD, we were a bit surprised, was not different between groups. Other studies have shown that RHFMD is impaired in smokers, but these, again, smokers had a very short smoking history, and we think that that's why this might be. But we were able to detect endothelial dysfunction when we looked at SSFMD. So this is our non-smokers had a larger dilation response to the sustained increase in shear stress compared to the smokers. So this would suggest that just measuring RHFMD might miss the presence of some dysfunction. So we performed a very similar study, this time comparing RHFMD and SSFMD in control and obese males who are otherwise healthy and young. And we saw, once again, we didn't detect a difference in RHFMD, but once again, the SSFMD was impaired. So this is our obese males with a smaller SSFMD response compared to our controls. So taken together, then this suggests that these two different sort of chronic conditions impair SSFMD, but dysfunction was not apparent in response to a transient increase in shear stress. We also look at acute interventions in my lab, and so the two that I'll end with here are talking about acute mental stress and exposure to oscillatory shear stress. So the stress task that we use in my lab, people come in and we have them do a speech in front of an audience. It's also videotaped, and then they have to do math out loud. So they have to do some arithmetic. I don't do it. Okay, so we looked at RHFMD and SSFMD, and this has been shown by other groups as well. We found that RHFMD was impaired about 15 minutes following exposure to acute mental stress. And again, this has been found by others. But when we looked at SSFMD, it wasn't impaired. So mental stress didn't seem to have a deleterious effect on SSFMD. And in a follow-up study, we sort of tried this again, and SSFMD was actually improved about 15 minutes following exposure to acute mental stress. So a really opposite response of RHFMD and SSFMD to this stimulus. And so lastly, we've looked at exposure to oscillatory shear stress. So this is just sort of a resting control Doppler velocity pattern in the brachial artery. And this is one with an established model that increases the amount of retrograde shear stress. So this is velocity below the zero line, so going backwards. So this is cuff inflation to about 70 millimeters of mercury placed on the forearm. It increases this backflow and also overall decreases the mean shear stress. And this shear stress pattern is sort of known to be deleterious and has been shown that with exposure, just 30 minutes of exposure to this oscillatory shear stress pattern pretty consistently results in transient RHFMD impairment in men. And so we performed a sex comparison. So we looked at men and women, and we found that after 30 minutes of exposure to this oscillatory shear stress stimulus, RHFMD was impaired. And so this was actually true in both men and women. In a separate study, we looked at SSFMD in men and women before and after exposure to this oscillatory shear stress. And we found that while SSFMD was impaired in men, it was not impaired in women, suggesting that there are some sex differences in vulnerability to endothelial dysfunction with oscillatory shear stress exposure that we were only able to detect by looking at SSFMD. So again, we're seeing these disparate responses where acute mental stress resulted in impairment of RHFMD, but actually maybe improvement of SSFMD, and that oscillatory shear stress exposure decreased RHFMD, but decreased SSFMD in men only. And so while it seems that there's utility in measuring SSFMD, I will just point out that the prognostic value of SSFMD has yet to be determined. And my panel is coming up here, and it's good because I'm on my last slide. So creating sustained controlled increases in shear stress in humans opens some new avenues of investigation rather than just being stuck with only being able to create a transient reactive hyperemia shear stress stimulus. SSFMD is a physiologically relevant response, so I think worth investigating. And chronic conditions and acute interventions appear to have a disparate impact on SS and RHFMD. So if we go back to this pie of endothelial functions and think about all these different indices, perhaps responses to shear stress can be divided in two here, so that when we're looking at transient and sustained increases in shear stress and the ensuing FMD, we're really getting at somewhat distinct parts of endothelial function. And so that means then that testing both RH and SSFMD might give us a more comprehensive understanding of endothelial function. And so I'll just thank all of my graduate students, former and current, that have been involved in this research, because without them, there is no data. So thank you very much.
Thank you, Dr. Pyke. And we'll take any questions now.
So I'm trying to put this in perspective in terms of, so, you know, I've been teaching medical students for a long time, probably incorrectly, but I've been guided by a colleague, Stefan Bolz, who I mentioned earlier, who would say, you know, shear stress is a very, very important determinant of, you know, regional blood flow in times of need, right, where valves have to actually dilate. He would say that large vessels, conduit vessels, the resistance is so low in those vessels that it's kind of like, you know, it may happen, but physiologically it doesn't need very much. I just want you to, like, can you put in perspectives, kind of like using Persuli's principle, for example, like how important is this in terms of, you know, really ultimately being necessary to get the right blood flow to tissues?
Right, and so that's a great point, because control of perfusion, when we're talking about dilation and changes in vascular resistance, that control is most important at the arteriolar level, so the vessels that are smaller than the conduit arteries. So how much the FMD in conduit arteries contributes to perfusion? Probably not so much, particularly in sort of a healthy model. I think when we're talking about the coronary arteries and we're talking about areas where there's a critical flow limitation because of a big lesion, there I think we sort of move that level, the point of biggest resistance up, right? And I mean, that's why we do angioplasty, to get rid of that plaque, to relieve the occlusion in the large conduit artery. So I think in those cases, and maybe in peripheral arterial disease where we have big blockages in the conduit arteries, improving the dilation to minimize the sort of impact of that big plaque, that might be helpful. So that's where FMD might be involved in perfusion when we're talking about at the level of the conduit artery. Otherwise, I think FMD in conduit arteries is really sort of telling us about the function of the endothelium there, that tells us about, again, the health of the endothelium, maybe not so much about its role in perfusion. And we do have to be careful, because the conduit artery endothelial function and microvascular endothelial function don't always agree. So we can't necessarily generalize what we would get in, what we would observe in a conduit artery to say that that's what's going to happen in an arterial.
And could it be like if you scale this down to like a mouse? This is why I've thought about it before, right? Once you scale it down to the mouse, that does start to become more and more relevant, right? And like most people have done these like isolated vessel experiments and whatnot, right? Like small rodents and stuff. Would you say under those circumstances it might actually be more important?
I would, right. So, I mean, certainly their conduit arteries are smaller, right? I mean, they're the size of our arterioles maybe. But I would still say that overall, because there's so many more arterioles and their capacity to dilate, again, I don't do animal research, so I'm really out of my depth here. But I would say, relatively speaking, the arteriolar dilation is still probably more important even when you have little conduit arteries, yeah, for perfusion, yeah.
I was wondering if you can control the shear, or as the blood flow velocity with your static hand grip. Is blood pressure response the same?
Right, so blood pressure increases a little bit when we do the isometric hand grip exercise. Again, it's rhythmic, so it's not like we're doing the sort of 2-minute hold and blood flow is limited to the arm. There's a big increase in blood flow in the arm. That's what we're sort of trying to achieve to increase shear stress in the brachial artery. But blood pressure does increase by 5-6 millimeters of mercury when we're doing the hand grip exercise compared to rest. In terms of compared to reactive hyperemia, blood pressure doesn't really change much when you do the reactive hyperemia test. Does that answer your question?
Yeah, I was just wondering if some individuals have a larger blood pressure response to actually achieve that flow velocity versus maybe others that actually have a pressure. But it seems like it's pretty small.
Yeah, and again, we test this really in young, healthy people. So whether there might, so there might be larger pressure responses in different populations. But I think, and that was also one of our concerns, that if we were getting big increases in blood pressure with this, were we activating the sympathetic nervous system? Could that be interfering with endothelial function? But we've done comparisons where we create the same increase in shear stress with heating, which would be passive, and we don't see increases in blood pressure. And so presumably there isn't sympathetic nervous activity increasing, and with hand grip exercise where we do see the increase in blood pressure, so we've got the same shear stress, and the dilation in the brachial artery, the FMD, is really the same. It's comparable. So it doesn't seem that the changes in blood pressure that we achieve with this hand grip model in healthy people are impacting the endothelial function or the FMD response.